Hello everyone. Let's go over lab exercise 2. In this lab, we are going to learn about blood. So what is the purpose of this exercise? Why do we want you to go over this? Because we want you to study the characteristics of blood and stimulate blood typing. And these are the learning objectives. So once you go over this lab exercise, please come back here and see that you have a good understanding of all of these concepts. Now let's go over some background information. As you know, blood runs in the vessels in our body and it has many important functions. One of the very important function of blood is transportation. It carries various substances to different parts of the body. So for example, it takes oxygen from the lungs and it takes it to the cells of the body, and from there carbon dioxide it enters the blood and it is taken to the lungs, so it can be expirated. Similarly, in the intestines, the nutrients, they are absorbed into the blood and then they are taken to different cells of the body. And nitrogenous wastes, they also enter the blood to be taken to the kidneys to be excreted. And as we already learned in lab exercise 1, that hormones, they are chemical messengers which travel in the blood. So blood has many important functions. And then the second important function of blood is regulation. It maintains the temperature of the body. It also maintains the normal pH. The pH of the blood, it is maintained in a specific range between 7.35 to 7.45. And why do we need that pH? Because if the pH is too low, the proteins, they will be denatured. Okay, because blood, it contains many substances. The plasma, blood is composed of plasma and the formed elements, which are the cells. So it contains many substances. If the pH is not managed, those components of the which are present in the blood, they can be damaged. How does blood do it? There are certain substances in the blood which are known as buffers. What are buffers? Buffers, they resist a very big change in the pH, right? So in this way, they maintain the pH in a certain range. Then blood also maintains the fluid volume in the circulatory system. And how does it do it? It contains many plasma proteins. Those plasma proteins, they in a way they attract, they keep the fluid inside the veins, inside the vessels. When plasma proteins are present inside the vessel, the fluid or the aqueous part of the blood, it stays inside the vessels and it does not seep into the tissues. Then blood also helps in protection. It prevents the blood loss. There are certain substances in the blood. If there is, for example, just consider this, if there is a cut in a vessel, those substances, they make a clot, right? So in this way, the blood loss, it is prevented. And then also it helps in preventing infection. In blood, we have antibodies, complement proteins, leukocytes, which are the white blood cells. All these, they fight infection. So one of the very important plasma protein is albumin. It makes most of the plasma proteins. The most common one is the albumin, and it is also a very important one. Let's quickly go over the composition of blood. Blood is a connective tissue, okay? And blood is composed of plasma and formed elements. Plasma is the aqueous part of the blood where gases, nutrients, ions, and hormones are dissolved. And what are the formed elements? These are the formed elements. These three components, they are the formed elements of the blood. They are red blood cells. They're also called erythrocytes. Erythro means red, cyte means cells. So erythrocytes is the other name for red blood cells. And red blood cells, as you know, they are disc-shaped cells. They're biconcave discs, and their primary function is to transport oxygen and carbon dioxide to the tissues. And they are biconcave disc, and please notice red blood cells, they do not have nucleus. We call it red blood cells are anucleate. Anucleate means that they are without any nucleus. Mature red blood cells do not have nucleus. Baby red blood cells, they do have nuclei. Nuclei is the plural of nucleus. As they become adult red blood cells, they lose their nucleus. So this is very important. Red blood cells, they do not have nucleus. And then you can see there is a lighter part in the center. It is because of their biconcave shape, because they lose the nucleus from the center and the hemoglobin is more concentrated on the sides. So this is how they appear under the microscope, and they are smaller in size compared to the white blood cells. So the second component of formed element are the white blood cells. They're also called leukocytes. Leuko means white, cytes means cells. And what is the primary function of the WBCs? They are immune cells, and they protect the body from foreign structures or diseased cells. There are many types of white blood cells. We will go over them in a little while. And then the third, okay, let's also look at this slide. So these are the white blood cells. As you can see, there are many different types of white blood cells. There are three granulocytes, and then there are two types of agranulocytes. So what does that mean? I will explain in a while. So our granulocytes are neutrophils, eosinophils, and basophils. And agranulocytes are lymphocytes and monocytes. So these are our white blood cells. And then the third formed element are the platelets. Platelets are also called thrombocytes. As sites again means cells, and why are they called thrombocytes? Because they are responsible for thrombosis. Thrombosis means like they make the clots. They are very important for clot formation. And platelets are not real cells. They are fragments of a cell that aid in clotting process called hemostasis. Hemostasis means that the blood is stopped. So now I want you to notice that white blood cells are the only cells which we call all the components of the cell. They have a cell membrane, they have a nucleus, they have a cytoplasm. Red blood cells, they do not have nuclei, and similarly platelets, they are only fragments. There is actually a very interesting way in which platelets are formed. When you are going to learn about it in your lecture. There are cells called megakaryocytes. If you observe these spider-like cells, these are known as megakaryocytes. And these processes, which are looking like kind of spider legs, these are the processes. These processes, they break. The pieces from these break, and they make platelets. They make these fragments, which are known as platelets. Okay. Now, so let's look at this. This I got from your Pearson e-text. Blood has plasma, right? This is plasma. When we centrifuge blood, plasma, which is the acellular part, it is present at the top, and 55% of the whole blood is plasma. It is the less dense. And then we have formed elements from this white stripe till the end, this red portion. Both of these, they contain formed element. This white thing is known as buffy coat. It contains leukocytes and platelets. So leukocytes and platelets together, they make about 1% of whole blood. Okay, and then this red portion, this is due to erythrocytes. We call this hematocrit. This is about 45% of whole blood. So normally this is hematocrit is 45%, and it comprises of erythrocytes or red blood cells. You have to know this value. You have to know this term, hematocrit. All right, so this is a slide. It is present in your Pearson e-text. I would like you to go to your text and read about different cell types. So I will quickly go over it. So erythrocytes are the red blood cells. They're biconcave, right? They're anucleate, they're salmon colored, and they're small compared to the WBCs. Their diameter is 7 to 8 micrometers. Okay. And then they are present 4 to 6 million per millimeter of blood. and their lifespan is about 100 to 120 days. And what is their primary function? They transport oxygen and carbon dioxide. So these are the red blood cells. Then we have white blood cells. Like I said before, we divide white blood cells into two major categories, granulocytes and agranulocytes. These are the granulocytes, granulocytes are called this because they have granules inside them. They have specific type of granules inside them. So first type is neutrophil. Neutrophil appears like this. It has multi-lobed nucleus. See over here you can see 1, 2, 3. It can be 1, 2, 3, 4, 5. It has multi-lobe nucleus. This is how we distinguish and recognize the neutrophil. You have to be able to recognize these cells under the microscope. So, and then the granules, they are neutral, right? The granules, they have certain colors, but in neutrophils, the granules, they are neutral. They neutralize each other. That is why they are known as neutrophils. And diameter is 10 to 12. Now compare this to 7 to 8 of the RBCs. So they are larger than the RBCs. When you will see a slide in which RBCs and WBCs are present, WBCs like white blood cells, the red blood cells will be smaller and the white blood cells will be larger cells. And what is their primary function? They phagocytize bacteria. They eat the bacteria and they kill the bacteria. Phagocytize means that they engulf it. They swallow the bacteria, they take it inside them, and then they destroy the bacteria. So their neutrophils are primarily, their function is to kill bacteria. Then we have number 2 granulocyte, which is eosinophil. Eosinophil has reddish granules. Okay, so you can see because of these reddish granules, the cytoplasm, it appears reddish, and it has bilobed nucleus. See, one lobe and two lobes. So eosinophil has bilobed nucleus, and size is almost the same as that of the neutrophil, 10 to 14. And what is the primary function of eosinophil? It kills parasitic worms. Okay, and it also plays a very important role in allergies and asthma. So with eosinophil, just remember parasites and allergies. Then we have basophil. Basophil have uh, uh, the granules, they are kind of dark and bluish. That is why the cytoplasm has a bluish coloration, and the nucleus is bilobed. It is not very prominent here because the granules are also dark in color. And what is the primary function? This is important. They release histamine. Histamine is a chemical which is a mediator of inflammation, and they also contain heparin. Heparin is an anticoagulant. Anticoagulant means a substance which prevents the blood from clotting. So these are our three granulocytes. Now let's come to the agranulocytes. We have lymphocytes and monocytes. So lymphocytes, they have a big nucleus. They have spherical or indented nucleus, right? And then they have a cytoplasmic rim. And they're a little smaller. The diameter is 5 to 17, and their main function is to mount an immune response. Then we have monocytes. Monocytes look at the size, and they have U-shaped or kidney-shaped nucleus, gray-blue cytoplasm. The diameter is 14 to 24. So these are the biggest of the cells. And what is the function? So our monocytes, they're traveling in the blood, then they diffuse out from the blood into the tissues. And when they go in the tissues, they change into cells which are known as macrophages. Macrophages, phage means to eat something, to take inside. So they are big eaters, right? So they also help fight infections. Monocytes, they are present in the blood. From the blood, they squeeze into the tissues. In the tissues, they are known as macrophages. And at different places, they have special names, these macrophages. Now our third formed element are the platelets. These are kind of fragments, but these fragments, they also contain granules. Those granules, they contain certain chemical substances which help in clotting. And what is their primary function? They seal small tears in the blood vessels, and they also start the process of blood clotting. So these are the formed elements of the blood. Now let's learn about blood types. As you know, we all have different blood types, and these blood types are based on two systems. We have ABO blood types, and then we have a rhesus blood types. So in this slide, let's discuss about the ABO blood types, and then we will go to the other one. According to these blood types, we have four blood types: blood group A, blood group B, blood group AB, and blood group O. So these blood groups, they are because of the presence of a certain type of antigen on the surface of red blood cell. What is an antigen? An antigen is a substance which has ability to induce production of antibody. So according in this blood type, the antigens which are present on the surface of the red blood cell, they can either be antigen A or they can be antigen B. And these antigen A and antigen B, they have the ability to produce antibody A and antibody B. So these antibodies, they are specific for their antigens. It means that antibody A, it can interact with antigen A, and it will only interact with antigen A. It will not interact with antigen B because antibodies are specific. Similarly, the antibody B will only interact with antigen B. Now now let's see how we get these four blood types. So in blood group A, the red blood cells of a person with blood group A, they have antigen A present on the surface of their red blood cells. They do not have antigen B. So we say we give this blood group the name blood group A because of the presence of this antigen A on the surface. Then in blood group B, the blood cell, they have antigen B on the surface. So we say these people, they have blood group B. Then in blood group AB, these people, they have both antigen A and antigen B on the surface of the red blood cell, and we say that this person has blood group AB because when I say the blood group is AB, it means that the red blood cells of this person or patient has both antigen A and antigen B present on the surface. And then we have blood group O. Blood group O lacks antigen A and it lacks antigen B. So there is no antigen A or B present on the surface of the blood group O. Now here is something important. Once we have settled how we name these blood groups because of the presence of the antigen. So now the way these genes are coded is such that if a person has a specific antigen, it will not have antibodies against that antigen, but it will have antibodies against the other antigen, the one which is missing. So for example, this blood group A in the plasma, it has antibody B. Antibody B is already present in the plasma, and it is floating in the plasma. So people with blood group A, they have antigen A on the surface and they have antibody B in the plasma because these people, blood group A people, their body does not recognize antigen B as their own, as self. It thinks antigen B is foreign. So they have antibodies to antigen B floating in their plasma. And now in blood group B, since they have antigen B and they lack antigen A, so in their plasma, they have antibody A. And now blood group AB, they have antigen A and they have antigen B. So they recognize both of these antigens as self. So they do not have any antibodies in their plasma. They don't have antibodies against antigen A, and they do not have antibodies against antigen B. And then in the blood group O, since blood group O, it is lacking any antigen on the surface, it recognizes both of these antigens as foreign, and it has antibodies to A and B in its plasma. So now let's move. The second system on the basis of which we type blood is known as rhesus system. So in this system there are many antigens. There are many reactive antigens in this system, but the most important is D antigen. And so if a cell it has D antigen, we say it is Rh positive, Rh for the rhesus system. And if it lacks D antigen, we say it is Rh negative. So according to these antigens, we either give a positive sign or negative sign to the blood. And just as I mentioned before, this Rh negative, it will have antibodies against antigen D. So it will have anti-D antibodies. Now merge the two systems. So we have these blood groups. We have A plus, A minus, then we have B plus, B minus, then we have AB plus, AB minus, then we have O plus and O minus. By minus I mean negative, and by plus I mean positive. And it is very important when we transfuse a blood, we have to make sure that the blood groups, they match. Otherwise, just like we learned, a person, for example, who has a blood group B positive, for instance, he or she has antibodies to A. So we cannot give a person who is B positive, we cannot give that person A positive blood. It will be, there will be a reaction of the antigens in the antibody, and it can be fatal. So that is why we have to be very cautious. Now, for example, just like I said, if our donor is A positive and a recipient is B positive, now what is going to happen? This person who is B positive, he has anti-A antibodies present in the plasma. So when this blood comes inside this person, these antibodies, they are going to attack antigen present on the surface of the red blood cell, and this can lead to a reaction which can destroy the red blood cells. And these antigens, they're also called as agglutinogens. Why? Because when these reactions occur, there is a process of clumping. These cells, they can clump together. For example, just imagine there is another red blood cell at this end, right? And then another, maybe the shape is not like perfect like this. It has many maybe processes, and then there is another red blood cell attached here. So these antigens, they make the cells clump together. That clumping is known as agglutination. And what is the benefit of this clumping? This clumping process aids in the removal of target by immune cells, because these antibodies, actually they are trying to protect this person from this foreign antigen. That is why they are attacking. This agglutination, it can help to destroy the pathogen quickly, but it can also be fatal in case of mismatched blood. Now this is the activity 1, and it is going to cover the learning objectives from 1 to 3, and you please read the directions and use the tables mentioned here. Go over these and then do these activities. So this is the activity 1, 2, 3 are name the three major components of blood, their role and percentage of composition, like we already did, the three major components of blood. And then name the formed elements. Name the following cells on histology slides and their key characteristics. So you have to be able to recognize erythrocytes, neutrophils, eosinophils, basophils, lymphocytes, and monocytes. We already went over this. Now this is how the slide is going to appear under the microscope. Now let's apply our knowledge, what we learned. So these pale and smaller cells with lighter centers. See, the centers are lighter compared to the rim, and they are smaller compared to these cells, and they do not have a nucleus. These are red blood cells, and red blood cells, they're present in more number compared to the white blood cells. See how many red blood cells you can see on this slide and how many white blood cells. Okay, so red blood cells, they are biconcave disc. That is why they appear lighter in the center. They do not have a nucleus. Adult mature red blood cells do not have a nucleus. And their primary function is they carry oxygen and some carbon dioxide. And then the second formed element is WBC, or white blood cells. So white blood cells, they are larger compared to the red blood cells. And there are many types of white blood cells as we went over. The one shown over here is neutrophil because it has many lobes. You can see clearly here 1, 2, 3, 4, 5, kind of 5 lobes in this cell. And the cytoplasm is kind of neutral, is it not? It is not very blue or very pink either. So this is a neutrophil. But basically at this stage, what in this slide we want you to recognize that this is a WBC and this is a red blood cell. And if you we can see that this is a neutrophil. And then we have thrombocytes. Thrombocytes or platelets, they're fragments, just like it is labeled over here. They are fragments, small fragments. Now this is a slide I will not go over again. You can read it from your manual. This is a nice helpful mnemonic to remember. Never let monkeys eat banana. There are 60% neutrophils, then 30% lymphocytes, then monocytes are just notice this. From 30% there are 6%, then eosinophils are 3%, and basophil are 1%. You have to know these. And then like I mentioned before, leukocytes have two categories, granulocytes and agranulocytes. So our granulocytes are neutrophils, eosinophils, and basophils, and agranulocytes are lymphocytes and monocytes. So these two are agranulocytes, and these neutrophils, eosinophils, and basophils are granulocytes. And please learn their specific characteristics, learn the shape of their nucleus, learn the color of their granules, and learn their function, which white blood cell is effective against which organism and what is the function. So this is important that you memorize this chart. All right, so now here is under the microscope. This is how neutrophil will appear. Now let me orient you. These more in number and lighter pale, kind of pale red cells, they are RBCs. And then this comparatively larger cell with multiple lobes and no specific color to the cytoplasm. This is a neutrophil, right? Neutrophil, it is called a neutrophil because the granules, they are kind of neutral, they cancel each other. Then in this slide, you can see an eosinophil. Now again, orient yourself. These are RBCs with more pale centers and darker rims. These are red blood cells, and then this larger cell over here is eosinophil. It has two lobes. Eosinophil is bilobed, and the granules are reddish. Okay. And then in this slide, again you can see this is a blood slide. You can clearly see the RBCs. And then this is a basophil. Basophil has a bilobed nucleus, which is not very prominent because the granules are also blue. Now in this slide, this is a lymphocyte. Lymph, again, just these are the red blood cells, and this is a white blood cell. What type of white blood cell? Lymphocyte. Lymphocyte is an agranulocyte. It has a large circular kind of nucleus which can be indented, and then there is a small rim of cytoplasm. So this is how lymphocyte appears. Then we have in this slide we have monocyte. Monocyte, the nucleus is kidney shaped or U-shaped, and then there is kind of bluish cytoplasm. And these smaller cells are RBCs. You have to be able to recognize these slides. You can also find them in your Pearson e-text, and I also highly encourage you and request you to go to PAL and have a look at the slides. All right, then your activity number 2 is learning physiology of the blood. Over here in this activity, you're going to cover your learning objective 4 and 5. Learning objective 4 is identify standard ranges for red blood cell count, white blood cell count, blood volume, blood pH, and blood osmolarity. Why do we need to know the normal range? Because if we do not know the normal range, we won't be able to tell that this is abnormal range or this is a pathology or a disease, because in certain diseases these things they are changed. And then you have to identify disorders associated with blood abnormalities. Now these are the instructions. You are supposed to use information in the table 2, and then you're supposed to answer the questions in the lab exercise work. This is the table 2. In this table, you can find values, normal value ranges of WBCs, RBCs, blood volume, blood osmolarity, blood pH. You have to learn these. And why is it important to know the normal values? Because if we do not know the normal, we will not be able to diagnose a pathology. In different conditions, these values change differently. And now let's go over part P, blood pathophysiology. They have started with the pH. So the normal pH is between 7.35 and 7.45. If it is below 7.35, we call it acidosis. Okay. And if it is more than 7.45, we call it alkalosis. And you can always, when in this course you will go over acidosis and alkalosis, you will learn further about it. So next pathophysiological terms we want you to know is leukocytosis and leukopenia. These are both conditions of the WBCs. Leuko means white, cyto means cell. Leukocytosis means too many white blood cells. So increased white blood cell count above the normal, and it can be indicative of a disease or inflammation. For example, if someone has inflammation, they will have too many neutrophils in their blood, and there too many neutrophils in the blood is going to increase the number of the WBCs, and we call it leukocytosis. The physician, when he gets the blood report, having a look at the blood report, the physician can tell that this person has some infection in the body somewhere because there are too many white blood cells. So the term is leukocytosis. Then there is leukopenia. Penia means deficiency of something. When there are decreased white blood cells in the number is in the blood, we call it leukopenia. People with AIDS, they have low number of white blood cells. That is why they get infections very easily. Actually, it is those infections which end up killing them. So the defect in AIDS is that those people, they have low number of white blood cells. Now, what are the terms related to the RBCs? We have polycythemia. Polycythemia means increased number of RBC. So the RBC count is going to be greater than normal range. And just a spoiler alert, these people will have a larger hematocrit, more than 45%, because hematocrit, RBCs make the hematocrit. And it can be indicative of types of cancers or other non-disease causes. What can be non-disease causes? For example, increased exercise, high altitude, dehydration. So these people also have increased number of RBCs. Why? Because when people who regularly exercise, they need more oxygen, and then they make more carbon dioxide, which needs to be brought back. So these people, they have more RBCs, and at high altitude, the atmospheric pressure of oxygen is low. It is compared to lower places. When we go to the high altitude places, the atmospheric pressure of oxygen is low. So to compensate for that, in long term, body starts making more RBCs. So oxygen can be delivered to all cells of the body. So these people also get polycythemia. Then what happens in dehydration? In dehydration, the fluid portion, the aqueous portion of the blood is low. As a result, the cells, they are concentrated. So that is also known as polycythemia. Then what is anemia? Anemia is decreased number of RBCs or hemoglobin lower than normal range. And there are several causes of anemia. It can be inadequate erythropoiesis. Erythropoiesis means that synthesis or production. So red blood cells are produced too little. Okay, so one reason can be too little production of RBCs. Second reason can be blood loss. This is known as hemorrhagic anemia. And the third reason can be RBC destruction. The body is making RBCs, but those RBCs, due to some reason, due to some pathological process, they are being destroyed. This is known as hemolytic anemia. And now this is activity number 3. It is blood typing, and the learning objectives are you have to define antigen and antibody. Please go to your textbook to find the definition, and then you have to outline the process of blood typing and antigen testing for ABO and RH blood type classifications. And for this, we will want you to go to your virtual lab simulations. So you have to complete these virtual lab simulations on Physio X part A and B. Please do it. And if you need any help, there is Appendix A and Appendix B at the end of each lab exercise. You can go there and you can see how to go to these sites. All right, so there is this virtual lab simulation, and please follow these steps and complete these activities. And when you are done, please complete the lab exercise worksheet. And this is then for blood typing. Thank you very much. So I'll see you or meet you virtually in next lab.